So today we're going to look at the organization of the skeletal muscle, and also we're going to examine the role of calcium in the muscle contraction and look at the mechanism behind a muscle contraction, which is the sliding filament mechanism. So if we look at how skeletal muscle is organized, it consists of multiple bundles of muscle fibers, a muscle fiber being the muscle cell. And inside each muscle fiber or muscle cell, we see multiple myofibrils. And the myofibrils consist of myofilaments that are both thick and thin. The thick myofilaments are myosin filaments, and the thin myofilaments are actin. And microscopically, we can see the light and dark bands or striations of skeletal muscle, which what we're actually visualizing there are the bands that make up the various sarcomeres, a sarcomere being the functional unit of the muscle. Now, a motor unit is a motor neuron and all of the muscle fibers or muscle cells that it innervates. And when a motor unit is activated, or when the motor neuron has an action potential that it carries to the muscle, all the fibers that motor neuron has the potential to activate together. So this is an all or nothing contraction where the action potential that's carried by the motor neuron to the muscle affects all of the muscle fibers that it's associated with. Now, one of the ways that the central nervous system controls the strength of contractions carried out by the muscles is by adjusting the number of motor neurons that fire. When more motor neurons fire, we see an increase in the number of motor units that end up contracting. And this is actually ordered that the lowest level of recruitment is going to involve motor units that have relatively few muscle fibers associated with those neurons. And then as more and more motor units are recruited, these are going to be motor units that will consist of more muscle fibers per motor neuron. Now, the frequency of the action potentials carried by motor neurons also contributes to the regulation of muscle tension. If two separate nerve impulses along the same motor unit happen close enough together to each other, what you have is an additive effect, where the first stimulus would generate a muscle twitch of a certain strength, and if the second stimulus takes place before a full relaxation of that motor unit, the second stimulus would produce a stronger contraction. And we can see that picture on this slide where we can look at the graph where we show time along the x-axis and amplitude of muscle contraction or strength of contraction on the y-axis. We see that if two stimuli, noted by the upward facing arrows along the bottom, are far enough apart, it triggers two separate muscle twitches of the same strength. However, if those two stimuli happen close enough together or close enough one after the other, we see the additive effect of summation. And then if the frequency of contraction is great enough, we can actually see tetanus take place in the muscle or the motor unit, which is a sustained contraction with no relaxation between twitches. Keep in mind that typically sustained contractions are actually due to asynchronous firing of different lower motor neurons, which then provides a steady level of input to the muscle overall. So even though there are multiple twitches taking place in individual motor units, this asynchronous firing of multiple motor units kind of masks that variability. So next we're going to take a closer look at what happens in the muscle during a muscle contraction, but we're going to do that by first looking at what takes place at the axon terminal of the motor unit. And I'm going to go ahead and insert a textbook video or a video from the textbook publisher to take a look at that.
An action potential arrives at the presynaptic terminal, causing voltage-gated calcium ion channels to open, increasing the calcium ion permeability of the presynaptic terminal cell membrane. Calcium ions enter the presynaptic terminal and cause vesicles to release their neurotransmitter acetylcholine from the synaptic vesicles into the presynaptic cleft. Diffusion of acetylcholine across the synaptic cleft and binding of acetylcholine to acetylcholine receptors on the postsynaptic muscle fiber membrane causes an increase in the permeability of ligand-gated sodium ion channels. The movement of sodium ions into the muscle cell results in depolarization of the postsynaptic membrane. Once threshold has been reached, a postsynaptic action potential is generated and is propagated over the muscle cell membrane.
So following that action potential in the motor neuron that triggers the secretion of acetylcholine, which triggers then the membrane of the muscle cell to become depolarized, triggering a muscle cell action potential. This resulting depolarization or action potential is conducted down structures called transverse tubules or T-tubules. And you can see that with the long, slender arrow that's going from the neuromuscular junction down the green tubes pictured. And when that action potential migrates down the T-tubule, it triggers the sarcoplasmic reticulum, which are those purpley or purplish, bluish structures adjacent to the T-tubules to release calcium ions. And it's these calcium ions that actually trigger the muscle contraction. So what we have is calcium playing a role between the excitation or the stimulus of the muscle by the motor unit being translated into a muscle contraction. And let's go ahead and use another McGraw-Hill video to link the process we just talked about with the next step.
At the T-tubule, the action potential is propagated on the T-tubule membrane into the interior of the muscle cell near the ends of the sarcoplasmic reticulum. The depolarization of the T-tubule membrane causes nearby voltage-gated calcium ion channels on the sarcoplasmic reticulum to open, resulting in an increase in the permeability of the sarcoplasmic reticulum to calcium ions. Calcium ions then diffuse from the sarcoplasmic reticulum into the sarcoplasm.
And let's go ahead and take a look at a few of the components of the sarcomere, which is the smallest functional unit of the muscle or the unit of contraction. And the sarcomere is arranged between two structures called Z-lines. And the one on the left on the upper picture is labeled, but you see that same structure repeats on the right below where the image is labeled I-band. That would be the other Z-line. Now, the sarcomere consists of thin and thick filaments, and during a contraction, what takes place is those thin and thick filaments slide in opposite directions, which shortens the muscle cell, which overall, as this happens in various regions of the muscle in different muscle fibers, it shortens the muscle, causing a contraction. Now, the thick filaments are made out of a protein called myosin. They're comprised of several myosin subunits that consist of two polypeptide chains each wrapped around each other. And then each chain has a globular head on it. And that globular head is the myosin motor. It's the structure that's actually going to generate the force that's going to result in a shortening of the sarcomere. And the shortening of this sarcomere, along with the shortening of many other ones, is what's going to result in a muscle contraction. Now, the thin filament is made out of two chains of actin proteins that are twisted together. In addition to the actin, there is also a tropomyosin strand that is wrapped around the thin filament as well. The tropomyosin, what it does is it covers myosin binding sites that are on the actin molecules, which inhibit those myosin globular heads or myosin motors from binding to the thin filaments. And then troponin, which is attached to tropomyosin, is the site where calcium binds to initiate the muscle contraction. And then you're going to see in this next animation coming up that when calcium binds to troponin, the tropomyosin strand moves out of the way, exposing the myosin binding sites on the actin molecules. And then the myosin motors, through the energy gained from the hydrolysis of ATP, shorten the sarcomere.
During contraction of a muscle, calcium ions bind to troponin. This moves tropomyosin out of the way and uncovers binding sites for myosin on the actin myofilaments. ADP and phosphate are attached to the myosin head from the previous cycle of movement. The myosin heads attach to the exposed binding sites on the actin myofilaments to form cross bridges, and the phosphate is released. Energy stored in the head of the myosin myofilament is used to move the head. This causes the actin myofilament to slide past the myosin myofilament. The ADP is released from the myosin head as it moves. The bond between actin and the myosin head is broken when an ATP molecule binds to the myosin head. The ATP is broken down to ADP and phosphate, releasing energy, which is stored in the myosin head and will be used later for movement. The head of the myosin molecule returns to its upright position and is ready to bind to actin again. If calcium ions are still present, the entire sequence is repeated.
And this slide here is for your reference. It's going to be included in the slide set that I upload, just showing the cross-bridge cycle that takes place between the action of the myosin motor protein and the binding of the sites on actin and the myosin motor power stroke and how that involves ATP. And then this slide summarizes the relationship between calcium and a muscle contraction and how low calcium levels inhibits cross-bridge formation and causes the relaxation state of a muscle. And when calcium is released from the sarcoplasmic reticulum and concentrations increase, then we have binding to troponin, and myosin gains access to its binding sites, which results in muscle contraction. And this next animation is just going to show the shortening of two adjacent sarcomeres, just to provide a little bit more of a big picture view of what's going on.
In a relaxed muscle, actin and myosin myofilaments lie side by side. During contraction, the actin and myosin myofilaments interact. The actins are pulled toward the center of each myosin myofilament. As a result, the sarcomeres shorten.
So today we took a look at the organization of the skeletal muscle and also examined the role of calcium in muscle contraction and looked at that sliding filament mechanism involving actin and myosin and the cross-bridge cycle.